Thank you very much for inviting me to this. I'm sort of the outlier in this. Everybody else is like a real specialist. Because I'm at a university, I'm more of a generalist. So I apologize if you think I'm talking down to you at any point. It's just the way I understand this stuff. So some of the details I might be able to go into more, but this is sort of my take on it. And I also, you have to forgive me, I'm used to teaching medical students a lot, so I have to dumb it down for them in the nicest possible way. But again, it's probably just the way I understand it. So what I'm going to talk to you guys about is understand how a transporter, that's how BioIVT, we got involved with them to try to track down this transporter, allows an acetylated amino acid. This is the acetylated amino acid. It's just leucine, N-acetyl-leucine. And it became, if the stars align, we're going to get an FDA approval on this probably by about September. So you wonder, well, how something like this could be an actual drug, and why doesn't leucine itself work? Those are sort of two of the background questions. So I'll walk you through that, and it turns out a transporter is very heavily involved with this, and that's why BioIVT was useful, and that's why I'm here. So as was said, this is me. I'm at Oxford. I have a professor hat on, and then years ago, I'll tell you more about our company in a little bit. This whole thing is going to be a narrative rather than just hard data and a lot of facts. Again, it's the way I understand the world. We co-founded a company because we were showing all our interesting basic research to big pharma, and they were disinterested, completely disinterested. So we decided to try to put our money where our mouth is and start a company. And it's been really challenging, like I say. I'll say a little more about that later, but it was a way to go forward. Another thing that drove me to this is both my parents. I grew up on a farm in Canada. That's the agriculture degrees. Initially I was going to farm, but the economy was bad. Both my parents, they're dead now, but when they were alive, they thought I was an embarrassment. I was still in school. I didn't make anything of my life. Now I'd be able to tell them we're actually trying to do something. We're trying to take our basic science and move it into drugs. So that's sort of in the back of my head. I'm probably getting too Freudian here, but that's another thing that's driven me towards this. So the outline of my talk, I'm going to talk about our goal, which was to get an FDA-approved drug. We haven't forgot about the EMA, and despite us leaving with Brexit, there's the NHRA. We'll do those, but this is where the most money is, so that's where we want to target first. Talk about our startup company, which we call IntraBio. Notice I inserted capital B here, just like any good company always puts capital inside their name. I'll talk about mechanism, and then pharmacokinetics, stereochemistry, and that got us to transporters. And then I'll give a little epilogue where the company is right now. So that's the overall strategy. In terms of our first goal then, we want to get a drug approved by the FDA. So that's a huge goal, but you've got to break it down into steps. So this is the US FDA approval. And we needed a disease, and it'll become clearer in the next couple slides why we chose this. This disease we abbreviate NPC. It's called Niemann-Pick C. It's a lysosomal storage disorder. So if you remember to basic biology, within cells, you've got the lysosome. It's where big macromolecules go to die. They get chopped up. If you have enzymes that don't chop them up, the lysosome just starts swelling. And eventually it's like if you live in a house, and if back in your student days, if nobody took out the recycling or the rubbish, eventually the whole household stops working. The lysosomes swell and multiply because they can't get rid of their cargo, and it affects them. Neurons are particularly susceptible for this. So in this particular disorder, it's a recessive allele, and it's a cholesterol or lipid transporter in the membrane, limiting membrane of the lysosome. And it's mostly manifested as neurological disorders, but there's also spleen effects, there's liver effects, mostly neurological, and mostly movement disorders, so ataxia. So I'll mention some of that later. And the drug that we were using for this is acetyl-L-leucine, what I showed you already. And the background to this is in France. I don't know if anybody here is from France, but this drug's actually approved in France. Jump ahead. Only approved in France. And it's sold over the counter. And you'll notice this is DL, this is just L. So the racemate is an over-the-counter drug in France. It's never been approved anywhere else in the world. So technically you could say what we did was a repurposing, drug repurposing strategy. And in France, it's sold over the counter for vertigo. There was a huge study done a few years ago by actually the guy that's with our company as a clinician, a neurologist from Germany. They showed it doesn't work for vertigo. But he was doing some studies in Niemann-Pick C patients for ataxia, and it seemed to work really well. So our company then decided we would try to translate all this through the one enantiomer. In the old days, we called it a chiral switch. We do clinical trials for this Niemann-Pick C, and we'd also do mechanism of action. And this is really appropriate. This is like my icon of an emotional roller coaster. This has been a crazy journey. We laughed, we cried, a lot more crying than laughter, unfortunately. So now I'll tell you a little bit about IntraBio. So it started in the Department of Pharmacology in Oxford. We actually have a standalone building, which is unusual. Usually you're just a floor somewhere. We've got a lovely building. It's a lovely place if you haven't been to Oxford. And this is me a few years ago, less gray, more hair, that sort of thing. I should update the picture, but vanity doesn't allow me to. This is Fran Platt. She's the current head of the department. These are the three scientific founders. This is Anthony Gallioni. At Oxford, you have professors of pharmacology like me and Fran. Then you have the professor of pharmacology. He's the professor of pharmacology, which he reminds us of once in a while. So I started with these people, and all our interests overlapped. I don't have time to go into huge details on that, but we're all interested in lysosomes in one way or another, and I was interested in small molecules. We used to take small molecules and try to make probes to look at signaling pathways. Then, like I say, about 10 years ago, I decided, why don't we try to do these for helping people? If Big Pharma's not interested, let's try to do the heavy lifting ourselves. Then we got a clinician involved, a neurologist at the University of Munich, Michael Strupp. And this is what really triggered things. So up to this point, we were trying to deal with, universities have tech transfer organizations, and they're supposed to help. It's almost like they do the opposite. They just slow things up and make your life miserable. They were offering us like $100K. You can't do drug discovery on $100K. They also wanted 50% of our company. And we negotiated for about 2 years, and we finally threw our toys out of the pram and walked away. Instead of a spin-out, we did a startup, and it pissed them off enormously. So we were really not liked by our tech transfer people in the university as a whole. But out of guilt, then the three academics gave the university back 3% of our shares of the whole company. So the university has 9%. Apparently, if you keep them less than 10%, they don't have a vote on your company. So that was out of guilt. But we gave it back. We got a startup. This guy is who really, everything's a double-edged sword. You notice him here? This is Fox News. He used to be a Fox News person. He was heavily involved in the Tea Party in the US. He moved to Oxford. He got involved with Anthony. It's a long story. But it's just been this tornado type thing, for good and worse and all this sort of stuff. And business people and money people think very differently about science than bench scientists, and that's been a hard learning curve. I bet we frustrate the hell out of them. They frustrate the hell out of us, but ultimately good things are coming from this, and I'll tell you that story. So now let's get into mechanism. So in terms of, I already told you about this, these observational trials. So these are almost case studies. So Michael Strupp noticed he was giving this Tanganil, depending on your pronunciation, the racemate of N-acetyl-leucine to patients for vertigo. But some of the patients had movement disorders and some had Niemann-Pick C. And they got better. And he has some kids in the Netherlands, 2 brothers that have been on it 12 years, and they're remarkably better. And the one that was younger when he went on it younger is better. This is a neurodegenerative disorder. So the median age of death is 11 years old. It depends on your genetic background, though. The same mutations, some people can live till their 60s or even longer. They'll be a little clumsy in this sort of thing. So the penetrance is very different with it. But it's mostly considered a disease of children, and some kids die when they're 2. So it's horrendous, and there's no drugs in the US to treat this. In Europe, there's a drug called miglustat, and Fran Platt, the current head of our department, discovered miglustat as an antiviral back in the day. But it actually works with this disorder. Now we're trying to do the same thing with this. So we knew the drug worked basically from these observational trials. You don't really know it works until you do double blind and that sort of thing. But we knew that it seemed to have a strong effect. So Fran then met this Michael Struck, who was doing these studies, at a meeting, and Fran's got what's called an NPC mouse. It has this transporter gene either knocked out or there's a spontaneous mutation that does the same thing. If you recall, it's a transporter for cholesterol or maybe lipids. The mouse really recapitulates a lot of the manifestations of NPC in people. So with another academic project, I've done a lot of work on psychiatric disorders, bipolar disorder in particular, and we use animal models there, and it is, I don't even believe in them, but you have to go forward. With this, it's a very good model, very good animal model for this disease, trying to figure out the mechanism, just out of academic curiosity, as I say here, purely out of academic. And then I came in, because I can do small molecule stuff, trying to look at some of the pharmacology. How does it get there? The pharmacokinetics, pharmacodynamics. Pharmacodynamics is a bit of a nightmare. I probably won't tell you much about that, but it's a metabolite. I can foreshadow it. N-acetyl-leucine gets cleaved once it's in a cell, and you get leucine. Leucine inside a cell is a powerful signaling molecule. Everybody thinks about mTOR. There's more to leucine than mTOR. A couple of its metabolites control flux through alpha ketoacid dehydrogenase, which is the same subunit as in pyruvate dehydrogenase, if you remember your citric acid cycle. And about 40% of pyruvate actually goes through what we think is the alpha ketoacid branch chain dehydrogenase. So we end up getting an increase in ATP from this. So leucine inside cells is powerful. It hits sirtuins. It's an allosteric regulator of glutamate dehydrogenase. It does a lot of things inside cells. So we basically have a prodrug and a transporters taking it across rather than leucine. I could end the talk there. That's sort of the take-home message, but I'll give you a few details now. So this is the opposite of the way people do drug discovery nowadays. Nowadays, you have a target, you validate the target, you take it through an animal model, and then it usually fails. We knew that it was doing something out here, and we went backwards. This is old school drug discovery. This is from like cannabinoids and morphine and this sort of thing, cocaine that was mentioned earlier. Plant-based products, you know it has an effect. Use Western medicine to try to figure out how. That's what we're doing here. It was just an academic interest. That changed dramatically. Once you start talking to investors and things, and big pharma, you need a mechanism. What's the mechanism? It's like it works. What do you care about the mechanism? It's academically interesting. I love this stuff, but what do you care? Almost everybody says the FDA requires it. It's a myth. If you look at the drugs approved by the FDA every year, there's one or two with unknown mechanism of action. It's not ideal, but it does exist. What they really say is it has to be safe and effective. So with that, mechanism can help these drug-drug interactions, which is a lot of your guys' fun, is very interesting for this. And you can make predictions, or maybe, what is it, the HERG, human ether-agogo related gene. Look at that. That will tell you something. But the actual mechanism in terms of how it's metabolized, I don't get that. Big Pharma keeps telling us it's for patient safety. Really, until you get it into people, you don't know about safety. You can make a lot of predictions, which is important. But if you overdo this patient safety and you don't get things taken forward, I think Brian used the example of clopidogrel. My take home was that you said nobody take that forward now. It's probably true. Nobody would have taken penicillin forward now because it kills guinea pigs. So we might have missed some drugs if we're a little over aggressive with it. We have to know what the safety profile is. Even COX-2 inhibitors, I try not to take too many asides, but I can't help it. COX-2 inhibitors I think are still a great thing, but they're eliminated. And investors, they always say safer investment because they don't read a lot. They just look at the bottom line. They look at whether you've met your primary output, and then they go through a set of tick boxes and they say, do you have mechanism, yes or no? I would argue for the diseases I like to work on above the neck, CNS diseases, I don't know if I believe in any of the mechanisms, really. Maybe dopamine D2 antagonists for schizophrenia. Beyond that, it's complicated. SSRIs do a hell of a lot more than just inhibiting the transporter. Neck down, fair enough. Target-based drug discovery works really well, and you can work out mechanism. So they argue safer investment. I don't know. The real reason for us then was, if you guys, everybody's younger than me here. There was a thing in like the 70s and it was Watergate, and there was a guy who kept telling these reporters, follow the money, and his code name was Deep Throat. If you follow the money, that usually gets to the bottom of anything in this or life. We had to work on mechanism not just because we were interested, but because of the investors, and if we're trying to get interest from big pharma, we'd like to be bought out. We don't have to be. We've got an office in the States now. We want to start selling drug in September if the FDA approves us. But we've got sort of two tracks. But everything is about the money when you get to this. So now I'm going to talk a little bit about pharmacokinetics, which I found odd. A lot of you probably won't find odd because you think about this stuff a lot. I'm an interested amateur in a lot of the stuff I'm talking to you about today. And we did this study and we got some weird results. But just first, again, it's for me more than your benefit, just to orientate myself. Why do we do pharmacokinetics for ADME? So this is plasma. You've already seen some of these curves. This is time. This is absorption on the way up. It hits a maximum, then it goes down. Elimination. So it'll be metabolized or peed out or pooed out or this sort of thing. So what I'm going to be most interested in is the Cmax. What's the concentration it hits in the plasma? And the area under the curve. What's the exposure to the drug? And we were comparing in these mice studies oral Tanganil versus our drug IB for IntraBio 1001. We started numbering it 1,000. Apparently it sounds better than just calling it number 1. I don't know why, but people wanted it that way. So we're doing the racemate versus the single enantiomer, the L form. L amino acids, remember, are the biologically active ones. And in our studies of this, pharmacodynamically, the L is the active isoform. And sorry, but a competitor of your guys' did this work. They did our IND package and that sort of thing initially. So they dosed the drugs with our racemate. This was not Tanganil. This was API grade racemate that we got synthesized. And you look here, this is in normal units, this isn't logged. You do get a little L, which is the biologically active one. You get a huge amount of D. These were equal. We ran them over a chiral HPLC. The areas under the curve were the same. It's the same amount. And yet we're getting a huge amount of D, very little L. So then we did just the pure L. You get a little D just because it's a contaminant. You can't get it absolutely pure. You look at the Cmax here, it's around 15,000. Over here, you can't really see it. It's about 3,000. So we had about a 5-fold increase in Cmax. And it recapitulated with area under the curve as well. So something was going on there. And if you're seeing differences between enantiomers, I'm jumping ahead a little bit, makes me think receptors. Going into this, I naively thought N-acetylation just probably speeds up passive transport across a membrane. I wasn't thinking about transporters at all. I was thinking sort of like N-acetylcysteine that everybody takes. You give it intravenously if somebody's overdosed on paracetamol. It's just a way of getting more cysteine into a person. So that's what I was thinking. So this was new to me. You guys probably look at this and think it's obvious. It wasn't so obvious to me at the time. And does anybody know why I chuck in a bunch of grapes at this point? How does that relate to stereochemistry? I'll show you in a second. I do this to Oxford medical students because it's a tough audience and you've got to keep them awake. So apologies, this leaks through as well. Sorry? Yes. Now you've given my punchline away, but yes. So with stereochemistry, just a little background on this. We think about it very much these are chiral molecules. So we've got a carbon here with 4 different groups arranged differently in space of the stereochemistry. And you can see that with a mirror. These are mirror images. This actually goes back in chemistry. So light, we'll think about light as just a wave. We'll leave the particle stuff to the physicists. It bounces up and down as a sine wave. The sine waves are in all directions initially. And then you can put it through a crystal. And there is something called Icelandic spar. And because of the crystal structure, it would allow just the light to go up and down. If you have a rope and you're flinging it between 2 people in a fence, you can only get waves to go up and down between the fences. That's what this is doing. If you go sideways, it cancels it out. Then you get plain polarized light. And then you run it through a solution, and it will rotate the light to the right or left. So that's optical activity. Then Pasteur here, well done with that. He was looking at this. And these are, if you guys drink wine, you've ever noticed crystals in it? It's tartaric acid. And there's different forms of tartaric acid that can crystallize out. A lot of my friends back in Canada think, oh my God, there's glass shards in the wine. And I try to educate them, and every time I go back, they say the same thing, so they don't pay attention. But anyways, these are tartaric acid crystals. What Pasteur did, between wine and between some purely chemical synthesis, he got sodium ammonium tartarate. And these crystallized into two different crystal forms. That's very unusual for enantiomers. And they were mirror images. So Pasteur looked down a microscope and just separated them all, and then put them into solutions, and it changed the angle of the light. Before that, everybody thought it was crystalline properties. He showed that it was a very property of the molecule. It makes sense now that we know what chirality is. So I thought that's a neat aside with this. Why is this important to pharmacology? Biology is chiral, so we have alpha-L amino acids, we have D carbohydrates. There's really cool ideas about that in terms of origins of life and that type of thing. I don't have time to get into, but if you're staying for the happy hour, I can bore you to death with some of this. You need 3-point binding. If you're going to distinguish between handedness, you need 3-point binding. And I've forgotten the names of the two people that put forward that hypothesis, but this affects receptors, enzymes, transporters. All these are made of alpha-L amino acids. A lady in the States at Stanford, I think it was, she made a completely D amino acid enzyme for carbohydrates, and it worked on L carbohydrates then. So that's really cool. It's probably just an accident that we're all alpha-L amino acids. This relates to the stereochemical differences that I was seeing in the pharmacokinetics. It suggested receptors, enzymes, or transporters, because we were looking at pharmacokinetics, turn to transporters. Just before I get onto that, consequences of stereochemistry. This is thalidomide. It's a poster child, evil poster child for everything that can go wrong with stereochemistry. So you can have fantastic stuff like wine and how that derived, and then something really tragic here. This is a stereocenter, and it's an acidic proton here. If you look here, these electrons can jump up, that proton will go off. It'll racemize in solution. And unfortunately, this was given as a sedative. In particular, and it was also anti-nausea, so it was given to pregnant women. And if you hit, I think it's like a 9-day developmental period, you end up with this really devastating disorder called phocomelia seal-like disease. The long bones in your arms don't extend, and you'll also not get proper finger development in these. And I'm a little younger to see this, but growing up, there were a couple people that had this. And they got so adept at using their feet for everything. I saw, well, I didn't see, it's complicated, friend of a friend. He saw this one guy take his foot, reach up, go into his inside suit pocket, take out change, put it into an old style parking meter and turn it. He drove a car that wasn't adapted for a handicapped person. He just had such hip flexibility and dexterity. Truly amazing, but now they've all got arthritis and things because these joints aren't supposed to be used like that. But this required the FDA changed their mind about some of this stuff and said you really have to do teratogenetic testing at that. So it was a big change. So everybody's very interested in stereochemistry. The FDA's rules now are, if you have a racemate, what the hell are you doing? You separate them and tell us what each one does and give us that drug. There's still some drugs that are grandfathered in, and just something that happened fairly recently. I teach pharmacology to medical students, as I said. We talk about salbutamol. I just had something like salbutamol, a performance-enhancing drug, before I got up here, because I've got asthma. And this hits the beta-2 adrenergic receptor. It's sold in the UK as a racemate. Our son had RSV, which is a respiratory syncytial virus. He wasn't getting enough oxygen. They gave him a nebulizer with salbutamol. His heart rate skyrocketed. Beta-1's on the heart, beta-2's on your lungs. You can remember that because you have one heart and 2 lungs. It's not supposed to hit that receptor, and yet it clearly did. So I've got to revise what I'm teaching now. It's all theory. Biology is very unforgiving like this. We're not like theoretical physicists where we can just write it down and nobody can test it, like string theory. We've got to be able to test it, and you get some surprises with that. So it's still really important, stereochemistry. So this is where BioIVT came in. So the first thing I do is you look at reviews. You Google reviews. And oh my God, there's 450 members of just solute carriers. Then there's the other families that are ATP dependent, ATP binding cassette ones. And it's just like, where do you even start with 65 families? So which one? If you're a big pharma, they can have, this is a robot, they can screen 10,000 compounds in a day, a lot of assays, they just throw money and resources at it. This is us, one man and his dog. So how do we ever approach this? Then I did other Google searching, and BioIVT popped up, and this is one of the things I saw, and I thought, that's a lot of transporters expressed. We want a bit of that action. So I emailed probably just like your help desk or something, and emails bounced around, and then work got done in various places, and I don't understand your company. It's everywhere. It's like, I don't know, like in James Bond, some of the evil, not that you're an evil organization, but Spectre, it's like everywhere. You just guys seem to be everywhere. So then, just a background on transporter analysis. If this is a membrane, lipophilic compounds just pass, they're passive. That's what I was sort of thinking about, just N-acetylation. We'll just make it passively permeate. If it's a transporter, there's a site of recognition. There's a binding event before it happens. And the experimental data shows this. So as you crank up the concentration down here, when you measure flux or rate or Vmax along here, you can treat transporters like enzymes. So this is effectively Michaelis-Menten. A transporter will show you saturation. Saturation, what I always teach the students, is the difference between biology and not biology. Pure physical chemistry is this straight line. If you get saturation, there's probably biology. Not always true, but probably biology. So this is what we're looking for in some of the results I'm going to show you. So we look at the chemistry and think about the biology and try to figure out what's going on. So N-acetylation, that makes like a peptide bond, and we get resonance, and Linus Pauling figured out alpha helices because this is planar, all these good things. So we thought, it must be a peptide transporter. You guys do PEP-T1, range the contracts and get that, and if anything, it goes down. So that's disappointing. So it wasn't PEP-T1. I convinced myself completely on this. Again, shows you biology is experimental, and no matter how good your idea is, the data will correct you. And that's where I like this line from Thomas Huxley, the great tragedy of science, the slaying of a beautiful hypothesis by an ugly fact. That's like the story. I should have that on my tombstone eventually. I'd run up against that a lot. So BioIVT was incredibly helpful, and a shout out to Mark Warren here, who I've never met any of you. I've exchanged emails with all of you, and it's really nice to put names to faces. But Mark I've never met. It's sometime I'd love to. I just did a search for his name, and I did a screenshot here. So Mark, if you guys don't know him, he's at the Santa Clara place in the States. When I first started interacting with you, I thought it was all European, and then this happened. And Mark was so helpful in so many ways. A lot of emails, being genuinely interested in trying to help us in the science. And he also had tremendous flexibility. We didn't buy, we could have bought compounds, get them shipped to the UK, and then try to ship them to the US. What a nightmare. Mark said, well, just tell us what you want. We'll order it for you and get it shipped to you. It seems trivial, but that made a huge difference to us in our project. And just even talking, some of the work we do is funded by the university and some is by the company. And the university is just a nightmare with dealing with this. Who are they? And they have to go through like 3 months of paperwork that you guys had to fill out to make sure it wasn't like money laundering and terrorism and all this sort of stuff. So it was so useful having Mark do that. And this one email I pulled because I thought it was very telling. And he said he checked with 2 most senior colleagues, so they were talking about this stuff. That's really cool that people in your company actually talk about this stuff. And I highlighted all the different transporters they thought it might be. And then down here, he says, so there you have it. 3 of us with over 60 years of experience can't even come up with a consensus guess for you. But he's lots of candidates, like I showed in that other slide. You guys have a lot of candidates if you want to go transporter hunting. Transporter hunting, I grew up on a farm in Canada, reminds me of this. So this, if you guys don't know, that's Elmer Fudd. All my comic examples are lost on the kids I teach nowadays. I have to explain it. So he goes hunting rabbits all the time. He was trying to kill Bugs Bunny. No rabbits were harmed in any of the cartoons. We were hunting transporters. So that's where we went for this. So we had another look at the chemistry. Again, you guys are going to say it seems obvious. It looks obvious to me now in hindsight as well, but it wasn't at the time. So if we look at the pKa of N-acetyl-leucine, we're down to just really a carboxylic acid at this point. Whereas the acetylation here drops the pKa to less than 0. Basically, it's not going to protonate. Whereas if it's leucine, it doesn't have this N-acetylation. You're around 9, sort of. Yeah, I don't have it there. I think you're around 9. So and what that does is you're basically an anion at all reasonable physiological pHs. Here's the intestine, here's most tissues. Lysosomes, it might be a little acidic, in your stomach a little acidic, but for the most part, if we're looking at transporters, it's going to be out here. It's an anion, so there's our carboxylate. So all those ones that Mark suggested, this MCT1 is a monocarboxylate transporter, so that's the one we put our money on there. It's the next guess. And we were successful with that. So it had a Km and a Vmax. Km looks high, but this is a metabolite. This isn't your grandfather's high affinity drug. This is a metabolite. Leucine itself will be contributing carbon skeletons to the citric acid cycle. It also, as I said, the metabolites will stimulate glucose production, glucose throughput. Rather than doing fermentation, glycolysis, and then making lactate, it will drive the mitochondria to use more of that glucose to complete oxidative phosphorylation. So this is sort of one of the endpoints of the story, is that the MCT, there's 14 family members. We just looked at one. When we write things and we talk to investors, we say it's good because there's 14 and they're everywhere. What we've got to do is start walking through them now and looking at individual ones and see what's going on. MCT1 does have a tissue distribution. So that human tissue, human atlas, we've looked at that. And there is some in neurons and some in glial cells, but there's number 2 and number 4, if memory serves, are more there. So we really should look at that. And what's happening is MCT is taking N-acetyl-leucine inside. Acetylase, probably acetylase 1, there's 3 acetalases that we know about, makes leucine. Then leucine basically gives you more ATP efficiency from glucose. And we've got data on that. We've even done some, with the clinicians, some PET scanning with FDGP. It's like fluorine 18 glucose, deoxyglucose uptake. So it just gives you a unidirectional amount of glucose uptake. It affects that. And we have data that it's important for, it looks like it's improving Parkinson's disease, Alzheimer's, restless leg syndrome. Before you think I'm just selling you snake oil, there's a lot of disorders that it doesn't work in as well. BioIVT, I told you the MCT story and the sadness of the PEP-T1 story. They also did we did some structure activity relationships to support a patent. We haven't published that yet. And this lactate, again, what I thought was a brilliant idea. These transporters can do exchange so that it would take lactate out and drive N-acetyl-leucine in. If a cell's undergoing lactic acidosis, what a brilliant thing. The very cell that's in trouble will be taking up your drug. Didn't work. If anything, N-acetyl-leucine blocked that. But that was MCT1. This is why we want to do a couple of the other isoforms of this. So now I'm down to sort of the epilogue. So where are we now with this? So we had a successful phase 3 it read out end of June. This was double-blind, placebo-controlled. It's a rare disease, so we only had 54 patients in it. That's how a small one-man and his dog type company can actually do some of this stuff. And the clinicians just like a month ago got this published in the New England Journal of Medicine, and the P was less than 0.05. I can rail on about frequentist statistics and things as well, but that's what they want. And investors, if it breaks that, they'll invest. If it doesn't quite break that, they're not interested despite the reasons. Don't like that, but that's how it got in there. And they got a glamour journal, and the clinicians get all the glory. We basic scientists think we've done all the heavy lifting. What do we get? We get a mechanism paper published in scientific reports, and we make sure that we say it's Nature scientific reports so that we can compete with the clinicians. A line from a singer-songwriter, Leonard Cohen. Maybe some of you know this. one of his lyrics was, well, never mind, we're ugly, but we have the music. So that's the way, metaphorically, I think about this. In terms of the FDA, we've got an NDA in with them, and they've approved it. So they haven't approved our drug, but they've approved that they won't kick out our NDA. So we've got a new drug approval sitting with them. We should hear about that June, July. And then we have something in the States that's called a PDUFA date. It's a prescription drug users fee, and that's in September. And I don't understand all the intricacies, but basically if we get it approved, we're going to be selling drug the day after that. And we have an infrastructure all set up in the States now. And we've got an office, rather than Oxford, we used to have an office in Oxford and a lab, but cost cutting, we had to shut it. We now have an office and lab, or maybe a lab, we're pushing for it, in Austin, Texas. So we're all ready to go on that. And then very personally, this is partly me, but mostly my wife. If this works, I'll get a little, they don't pay academics well. And especially in Oxford, it's very expensive. If this works and I earn a little money, we'll get a bigger house in Oxford. So in terms of what I've told you about, our goal was to get an FDA-approved drug. We might be that by September. I'll be laughing or crying. I told you about mechanism. I told you about how BioIVT was really important with this transporter idea and how that's integral with our mechanism. And for people that don't understand that, I tell them about L-DOPA. It needs a transporter. What you really want is dopamine levels in the brain. You can't do that directly. You need a precursor. And the epilogue was that we've got a couple, one glamour, one sort of son of a glamour journal publication, and we're in talks with the FDA. And I also want to just say, we used to get beaten up really, really badly. I told some people here with the FDA meetings, we've had like 8 meetings with them. We just used to come out bruised and bloodied. And after our successful clinical trial, they were friendly last time, and it had us all off guard, and it was silent after the meeting because we didn't know what was going on. They're never nice to us, and they were nice to us for a change. So thank you for your attention.